Welcome dear students to the online classes. Today I'm going to talk about applications of microorganisms. The previous lecture we had was about the harmful effects of microorganisms, and you might be aware now that it's only 1% of the microbes that are pathogenic for humans. So it means that the beneficial part is on the upper scale than the harmful effects. So let us begin today by talking about the applications of microorganisms. So microorganisms, they are by their omnipresence, microorganisms by their omnipresence, they impact our whole entire biosphere. So microbial life plays a prominent role in regulating our planet's entire biogeochemical systems. So you might be aware with the cycling of carbon, cycling of nitrogen, cycling of sulfur. So when you look on around yourself, so you see that microbes are found in diverse environments. They can be found in extremely frozen zones. They can be present in hot springs. One of the example I can give is Taq polymerase. Taq polymerase was isolated from a microorganism which was found in the hot springs, that is Thermus aquaticus. And today we have its application in PCR, that is polymerase chain reaction. We have its application in genetic engineering, which is the prominent and the cutting edge part of today's today's research. So they are also found in acidic lakes where the pH is below, pH is below 2. They are present in hydrothermal vents that are they are present at the bottom of the deepest oceans, and of course they are present in the humans. As I told in the last lecture, we are humans and microbes are inside us and outside us. So we have something called as skin microflora. One example I can give is S. epidermidis. S. epidermidis. It's present on our skin. Another, we have many of them in oral cavity. We have the gut microflora, one of the helpful bacteria you will find in your gut flora is E. coli. And we have many other microorganisms that are present in your different tracts. One example is urinary genital tract. Okay, so next let us see what this field is called. So when we talk about applications, we talk about a field that's called applied microbiology. So this is a field which contains many other fields under it. So this applied microbiology, it refers to the field which is not involved in any study as such regarding the microorganisms, but it is the study of certain processes and many things that are isolated from the microorganisms that are beneficial to the human beings. So we can culture microorganisms, and so that we can isolate things like antibiotics from them, penicillin, for example, isolated from the mold Penicillium notatum. Penicillium notatum. Okay. We have different kinds of enzymes, for example, polysaccharides, proteases, which are being isolated from the microorganisms. We have many kinds of polysaccharides like xanthan. Then we have different kinds of polyamides. We get many different kinds of amino acids from microorganisms. Two examples I will give here is vitamin B2, that's riboflavin, and vitamin B12, that is cyclocobalamin, which helps in formation of the red blood cells. We have different kinds of vitamins we can get from microorganisms. And now talking about two things I told you about, isolation and second is processes. So what kind of processes this helps microbes help in is fermentation, where you get ethanol when you use pyruvate as a source of starting material. In absence of oxygen, the yeast, that is Saccharomyces cerevisiae, can convert the pyruvate first into the acetaldehyde and then finally into ethanol. So we can and it's also used in brewing industry. In the wastewater treatment, we use different kinds of microbes that can gulp up the waste, and then they can also be involved in biodegradation, or you can say bioremediation of many domestic, agricultural, and industrial wastes. So bioremediation means it's kind of a remedy that microorganisms can degrade something which is not good for us. So I can give an example of oil spills. When oil spills happen in the oceans, then we can use microorganisms which use these hydrocarbons. Actually, they use these hydrocarbons and convert them into the non effective, non toxic things. So microorganisms have applications in different scientific fields. I can give example of it. We can inshallah start with the medicinal fields, because we are or you are particularly involved in the medicinal lines, paramedical staff. So we will see that they are also have applications in pharmaceuticals, applications in industrial microbiology. Then we have environmental microbiology, agricultural microbiology, aquatic and other kinds of different fields which have a huge economical potential. And to the top of that, adding the crown to the microorganisms, came the field called as biotechnology. We'll be discussing about it briefly after some time. So it has emerged as a new field, and this field has been used where the microorganisms are being used as we can hijack the machinery of microorganisms and make it to do what we want. So one of the best examples was insulin, that was first named as humalin. It was isolated from yeast when we genetically engineered the yeast by putting on the genes inside the yeast so that it can produce our insulin. Insulin, because it's only 52 amino acids long, so that was the first attempt that was made. And it means we can manipulate the genes within the microorganisms so that they can be beneficial to us. And recently it has also been proposed that microorganisms can be used in cancer therapy. Okay, so let's move to the next slide. First comes the medical microbiology. In case of medical microbiology, it's a branch of medicinal science actually that is concentrated with the prevention of any kind of infectious disease, its diagnosis, and its treatment. And there are four kinds of microorganisms that can cause infections in humans. We have viruses, currently we have coronavirus, and we have HIV virus. They are more prominent viruses. Both of them are RNA containing viruses. Then we have bacteria. For example, we have Staph aureus that causes MRSA, methicillin resistant Staphylococcus aureus, and which are drug resistant actually. And there are many kinds of fungal diseases like mycosis of skin. And then we have parasites. We know best examples are malaria. They cause malaria, and they can also cause filariasis. And there are also some kinds of proteins which are actually not are not able to form their proper three-dimensional structure. So what happens is when they are not able to act as a normal protein, it converts into an like a different kind of a protein, which is like of different shape, and it has been found to cause Alzheimer's disease in humans, which is what we mean by loss of memory with age. It also includes the study of pathogenic microorganisms and their role in human illness. So it means, in short, we will get a concept of microbial pathogenesis, how microbes can interact with our cells and make our cells or make them ill. So it also involves the study of epidemiology, means that where the disease is being more rampant right now. It talks about the pathology, how the microorganisms can damage our tissues, that is concerned with the pathology. And finally, our immune system, which is immunology. So immunology can divide into two things. One is when you have defense, your immune system first tries to defend itself from the microorganisms, and once if the microorganisms actually overwhelm your immune system, then they provide something called as response. So this response will come in the form of antibodies. Okay, and defense comes in the form of macrophages, in the form of neutrophils, etc. In addition to this, the field of science studies also includes various clinical applications of microbes so that for the overall and improvement of the health of the humans. This area of microbiology can cover other aspects also, like human microbiota. It will be talking about the details, how many different kinds of microbes are lining our gut, present in the large intestine, which are called as fermenters, in small intestine also. And talking then we can in medical microbiology we can learn about the cancer and tumor microenvironments. Means when you look at the tumors, you can see that what are the environments surrounding them. So in this case, in medical microbiology, we also see that what other organisms are present in the vicinity that can cause cancer, or it can, you know, may help in causing cancer. For example, viruses are the main cause of cancers. So if these viruses are surrounding this, the tumor environment, so we can study that also in this under this field. Okay, next slide. So we were talking about pharmaceutical microbiology. Pharmaceutical means production of medicines. So this study of microorganisms is associated with the manufacturing of various pharmaceutical products like anti-infective agents like antivirals, vaccines like BCG, the Bacillus calmette-guerin, that is given for tuberculosis for children, and there are other vaccines also. For example, polio vaccine is there. Okay, and related products like insulin, as I talked about, humulin, that was being genetically engineered for the production of human beings. Otherwise, earlier it used to come from the pigs and other sources. We also can produce now human growth hormone by the process of genetic industry. So, what are the things that come under pharmaceutical microbiology is production of antibiotics. So, antibiotics can be produced from fungi, and this penicillium notatum can produce your drug called as penicillin, which causes penicillin, which causes the breakage backbone, which causes the breakage of the backbone of the cell wall of both gram positive and gram negative, that is peptidoglycan layer. We also have phallosporins and a species of bacterium called as streptomyces. It comes from the actinomycetes, and many of the drugs have been isolated from streptomyces. The first drug that was isolated from streptomyces was which was effective against tuberculosis was streptomycin. Streptomycin. So you can see that many kinds of antibiotics have been produced from or isolated from the. So actually, if you concentrate on the term antibiotics, it means one organism is producing toxic substance against the other organism, and it's preferably okay, because what they are doing is they are trying to increase their influence in their environment. So they are producing such toxins that it will repel other bacteria or viruses. And many of these drugs are natural, means they are naturally isolated, or they can be then they can be either synthesized as in a chemical form once we know what it's made up of, or we can also keep them semi-synthetic, means we can get the isolate the natural antibiotic, and then we can add some derivatives to it so that it makes it more effective. And second thing that comes in the pharmaceutical microbiology is containment of contamination and spoils in by microorganisms. Or in other aspects, you can see that we have to see that we can minimize the number of microorganisms in the process environment of producing the drugs, and then also excluding the other aspects, byproducts of microorganisms like exotoxins, endotoxins, so that we ensure that the finished final pharmaceutical drug is very sterile. And then we talk about research and development. Microorganisms are being used to detect mutagenic. I mean, the ability of any cell to turn into the mutagenic can lead to cancer. And we also see that if we use these drugs, it should not be cancerous. So these activities can be done first on the microorganisms to see whether they are mutagenic or carcinogenic for that one particular prospective drug. So let's move on to the next application. We have industrial microbiology. In this case, the term industrial means in this way the term industrial microbiology, it refers to the use of microorganisms actually for the industrial purposes. And in industry, many things can be produced. For example, anticoagulants like heparin, antidepressants, and vasodilators, herbicides, insecticides. Herbicides particularly kill the weeds, and insecticides, as the name itself says, it repels the mosquitoes. We have plant hormones like different kinds of plant hormones like auxins, cytokinins. Okay. We can in industrial microbiology we can produce enzymes like amylase, which can degrade many kinds of small carbohydrates to form ethanol, and then other organic solvents that have been isolated or produced in large quantities by genetic engineering. As I was telling before, that either we can isolate the things directly from the microorganisms, or we can genetically engineer the microorganisms so that it becomes a factory, it becomes a cell factory for us to produce the things we want from them. So it's actually manipulating them. Okay. And what we get in industrial microbiology is we will get different kinds of nutrients, polysaccharides, enzymes. In case of nutrients, we can get vitamins like vitamin B2, that is, and then vitamin B12, that's cyclocobalamin, vitamin B2 riboflavin, and then B1 is riboflavin, and B2 is another kind of a B complex vitamin. We can have different kinds of amino acids. We can have lysine, like lysine, arginine, and histidine. They are basic amino acids, and from them different kinds of amino acids can be produced. So it is produced by a bacterium called as Corynebacterium glutamicium, and it's prescribed to treat herpes simplex infections. Means it is a viral infection caused by herpes simplex virus that can cause cold sores on your lips. Cold sores. Okay. Polysaccharides, we have xanthan. Xanthan is produced by the bacterium Xanthomonas campestris, is used to stabilize and thicken foods, and it acts as a base for the many different kinds of cosmetics like lipsticks and other kinds of face creams. Then in petroleum, and it also acts as a binding agent in many pharmaceuticals, I mean drugs, and it's also used in textiles, printing, and dyeing of different kinds of clothes. Another kind of polysaccharide that is very much coming from the microorganisms is from the bacterium Leuconostoc mesenteroids. When it grows on sucrose, it is able to extend the blood plasma. So I mean we can extract or extend the blood plasma levels. So then comes the company of issues, enzymes. So where bacteria can produce different kinds of amylases which are used in brewing industry, baking industry, and textile production. We have also proteases and lipases. Proteases are used for tendering meats, preparing different kinds of leathers, making detergents, and also for the production of cheese, which has been done way before the microbiology was discovered by different kinds of people in Roman empires and many other previous centuries. Now we also have one of the applications of, or we can say that one of the applications of microorganisms is environmental microbiology, which deals with the environment which we are living in. So this is the study of the function and the diversity of different microbes which make our natural environments, so that we can live in cohesion with them. So it involves the characterization of key bacterial habitats, different kinds of habitats where they live, and such as rhizosphere, which is combined with the plants, phylospheres, soil, groundwater ecosystems, open oceans, or extreme environments, where we call them as extremophiles. The extreme environments are like, as I told before, acidic, or it can be hot springs, or it can be completely deep freezing environment. This field also includes the branch of different microbiology, that is microbial ecology, where we study the relationship between two things, the relationship between the microbe and its environment. Okay. We have also microbiology mediated nutrient cycling. We have carbon cycle, where carbon fixation takes place by photosynthesis and then used by us while having digestion. Nitrogen cycle, where the nitrogen of the environment, which is very abundant in the air, is being fixed by the nitrogen fixing bacteria. Then we have sulfur cycle and methane metabolism, CH4, which is used as a cooking. It is used as a for cooking also as a gas. In environmental microbiology, we also have used it for wastewater treatment, as I talked about it before, and we can use it for clearing different kinds of oil spills if there are in the oceans. And it happened during the war between Iraq and Iran, when there was a lot of oil spilled inside the oceans, because of which many fishes were dying. Now, coming to the agricultural and aquatic microbiology, it is the study of relevant agricultural products, and it contains different subdomains like soil microbiology, where we study different kinds of microorganisms that are present in the soil. For example, microorganisms which are present in different kinds of soils, like sand soil, clay, etc. Then we also have, under this domain, we have plant microbiology, where we study the interactions of these microorganisms with different kinds of plants. For example, nitrogen fixing plants. They have applications in food microbiology, where we have to study which microorganisms are causing food spoilage, and what are causing the foodborne illnesses. For example, Clostridium botulinum. Clostridium botulinum causes food poisoning. Botulinum. Okay. In aquatic microbiology, we study these microorganisms that are found in water that cause different kinds of diseases in fishes, that can cause diseases in other marine sources of food. So they have a role to play in the agricultural and aquatic microbiology. Next comes the pinnacle of microbiology. This biotechnology is the pinnacle application of your microorganisms. So what does this term mean? What is this field all about? I've done my master's in the same field, so I can, as far as I know it, I have a definition of it. So what biotechnology is, let me define it first for you. So it is the use of modern sophisticated techniques. It includes PCR, it can include your microscopy, it can include very different kinds of techniques. So biotechnology is the use of modern sophisticated techniques to manipulate the biological systems. Manipulation comes from the gene level, okay? This is how we can manipulate the microorganisms. So biotechnology is the use of these modern sophisticated techniques like genetic engineering, restriction enzymes which you use in cutting up the DNA. So biotechnology is the use of these modern sophisticated techniques to manipulate the genes in different biological systems. It can be microbes, plants, or animals, or it can be us, for the benefit of the humankind in the form of products and services. So product example is insulin, okay, growth hormone, and services is like cleaning of wastewater treatment and cleaning of oil spills. So what we can do is we can genetically engineer microorganisms that can act on the hydrocarbons and make them non-toxic. Okay. So biotechnology is a broad area of biology. It means it depends on the tool and its applications. So it also combines with or overlaps with other scientific fields. So biotechnology has applications in four major microbial areas that include medical, which I already talked before on the first hand. That's healthcare. We also talked about the industrial applications of biotechnology, where we can enhance or we can broaden our production by using microbes which are genetically engineered, and we can also use them for biodegradation of plastics, vegetable oils, biofuels, and they can also be used in environmental and agricultural production, that is production of different kinds of crops. One example is BT cotton. BT cotton is a cotton which is not being affected by any insects. So this bacteria is called Bacillus thuringiensis. 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 Okay. So BT cotton, it's a genetically engineered cotton. We also have pomato. Tomato. Tomato is a combination of tomato and potato. That's called pomato. And definitely it's a little bit funny also. I like the terms which they use in biotechnology. So, how did this field emerge? It was in the late 20th and 21st century that biotechnology expanded and to include new and diverse sciences such as genetic engineering, what we today call as recombinant DNA technology. Recombinant means it's a recombination of two different DNAs from two different organisms. For example, bacteria contain plasmids. We can take the plasmid, cut it with a restriction enzyme, and then put our gene of interest in that, and then we can put it back into the bacteria. So the bacteria becomes a factory for our production of the things we desire. Okay. Now it has expanded. Now biotechnology has expanded from to study of the genomics and proteomics, applied immunology. Applied immunology means how can we produce monoclonal antibodies. Okay, and development of various pharmaceutical therapies using this technique of biotechnology. We can produce what is called as nanotechnology. Nanotechnology can produce small. This nanotechnology is a field where we can, put the drugs into very nanoscale particles that can have long-lasting effect. Okay, and then it's also helping us forming different diagnostic tests which can be very fast and very cost effective. For example, in case of coronavirus, we are in very much need of a cost effective diagnostic tool, so it can come from the field of biotechnology. So hope you all like this lecture, and please provide your comments and other whatever you want to ask me. You can ask me on our WhatsApp group, or you can just put a comment when I put it on the YouTube. So dear students, that's it. I will surely meet in the next lecture. Hope everything is well with you. Let's hope that and we get rid of this coronavirus. Assalamu alaikum.